Hello and welcome to the final Bio43 pre-lab video. This week, we'll conclude our MoBio unit with the expression and purification of our protein of interest, and start thinking about some of the experiments we could perform to learn more about it. Last time, we left off with our gene of interest cloned into the PET16B plasmid. Now we need to put this plasmid into E. coli cells, which will do the work of expressing the protein encoded by the gene. The process of bacterial cells taking up foreign DNA is called transformation. Some bacteria are naturally able to do this, but E. coli requires special preparation. A chemical treatment makes the membranes more permeable to DNA. Cells treated this way are called competent. We can mix the competent cells with our plasmid, and then shock them with either an increase in heat or a brief electrical pulse. This will disrupt the cell membrane enough for the DNA to enter the cytoplasm. Give the cells some time to recover, and that's it. But we're not quite ready to express our protein yet. The bacteria we have at the end of the transformation process will be a mixture of several different things. The vast majority of E. coli cells won't actually have taken up any DNA. This is why plasmids contain antibiotic resistance genes. PET16B has a BLA gene, which encodes a protein that grants resistance to the antibiotic ampicillin. So we take the transformation mixture, spread it on an agar plate with ampicillin, and only the cells containing a plasmid will grow into colonies. But not every colony will necessarily have the correct plasmid. Ideally, our gene of interest was ligated into the plasmid, but some fraction of the time, the plasmid will just reseal itself. Additionally, the restriction enzyme might just not cut every copy of the plasmid. So some of these colonies will just have regular PET16B without the gene of interest inserted. Another problem is that the gene of interest might not have been inserted with the correct orientation. If it flipped around like this, the RNA polymerase will not read the correct strand and won't make the protein we want. So we need to figure out which colonies have the plasmid we want and which have something else. A good approach for this is a diagnostic digest. This is a restriction digest like we did before, but rather than being a step in the cloning process, this gives us information about unknown plasmids. So we'll take the colonies we have here and purify the plasmid DNA from each one separately. Then we'll cut a portion of each sample with the same restriction enzymes and get different sized DNA fragments depending on which plasmid we're dealing with. To decide which enzymes to use for the digest, we'll return to benchling. Bring up the plasmid with your gene of interest inserted. Go to the digest menu, and change the settings to display only single cutters. That is, cut sites that only appear once on the plasmid. And we don't need to worry about ND1, ZO1, and BAMH1 anymore. We want to pick one site that is within the gene of interest and one that is outside of it. The site inside the coding sequence should be close to one of the ends, and not too near the middle. There are a lot of options here, but let's go with SAC1, since I know my lab keeps a stock of that enzyme. When you're doing this, you can assume you have access to any restriction endonuclease. The second enzyme you choose, the one outside the gene, should have a site about 1,000 bases away from the first. Though anywhere from 500 bases to 2,000 or 3,000 bases would be fine. If you click on the plasmid near the first site, hold down the mouse button, and drag towards other sites, you can estimate the distance between those sites. This BCL1 site is between 11 and 1,200 bases from the SAC1 site. Let's use this pair of enzymes. We can go back to the digest menu, only display selected enzymes again, and select SAC1 and BCL1. Now click Run Digest. The top table has some information about how to perform the digest. These are the temperatures each enzyme works best at, and these are various buffers the digest can be performed in. We won't worry about that part in this lab. What we really care about is this bottom table, specifically the length column. This tells us that after digesting with these enzymes, we'll have two DNA fragments. One will be 1,168 bases long, and the other 6,892. Now suppose we cut the original PET16B plasmid with the same enzymes. Go to that file, select SAC1 and BCL1 again, run the digest, and this time we only get one fragment with a length of 5,711 bases. This makes sense, since the SAC1 cut site was part of our gene's coding sequence and isn't normally found on the PET16B plasmid. Only BCL1 will cut the plasmid, giving us one long linear piece of DNA. Now we need to think about what size fragments of DNA we'd get if the gene inserted in the wrong direction. We don't have a benchling file for that now, but it's easy to make. First, make a copy of the PET16B plasmid. I'll call this PET16B MEFV flipped, then go to the file with your PCR product. Select the whole thing, create, new DNA, but tell it to use reverse complement instead. This is the same sequence as a PCR product, but with the orientation reversed. I'll call it MEFV PCR flipped. Then we'll recreate the ligation step just like before. Copy the bases from the starting zone 1 site to the ending site, go back to PET16B MEFV flipped, highlight the zone 1 site there, and paste. You can see it's flipped since the reading frame is pointed in the wrong direction. So now we do the SAC1 BCL1 digest one more time, and we see that we get fragments with lengths of 3,007 bases and 5,053 bases. So these plasmids, cut with the same two enzymes, would have very different results. But we need a way to see the DNA fragments after the digests. For this, we use gel electrophoresis. I've linked to a simulation on this, but in brief, the different digests are put into wells at the top of an agarose gel. Agarose forms this mesh framework. An electric field will pull the DNA towards the other end of the gel. But larger fragments will have a more difficult time moving through the agarose mesh compared to smaller fragments. So the smaller pieces will travel farther along the gel in a given time frame. To see how far the DNA traveled, we need to use a dye like ethidium bromide. Ethidium bromide binds DNA and fluoresces under UV light. In addition to our digested plasmids, our gel will also include a mixture of DNA fragments with known sizes. This is called a marker or ladder. By seeing how far our cut plasmid pieces migrate compared to these known pieces of DNA, we can tell the sizes of the plasmid fragments. Though these are just rough size estimates. This method couldn't help us distinguish, say, an 1170 base fragment from an 1190 base fragment. So you want to make sure your desired plasmid has at least one fragment that is easy to distinguish from the other possibilities. No fragments that are within maybe 20% of its length. This digest works well since the plasmid we want has a fragment of about 1170 bases, and no other plasmid would produce a fragment below 3000. It's easy to see that this banding pattern comes from the plasmid we want. Now that we have a confirmed stock of the proper plasmid, we transform it into E. coli again, and then it's time to express our gene of interest. Recall that our plasmid contains a T7 promoter where an RNA polymerase will bind and transcribe our gene. But we don't want to start the expression right away. Dedicating all that energy to making our protein will affect the growth of E. coli. So we want to wait until we've grown a lot of E. coli to begin production of our protein. That's where the LAC-I gene and LAC operator come into play. LAC-I produces a repressor protein that binds the LAC operator near the promoter and stops the RNA polymerase from binding and transcribing the sequence that follows. But if we add a chemical called IPTG, it will cause the repressor to stop binding the operator and transcription can occur. So we grow up a bunch of E. coli cells, add IPTG, and then the cells make a large quantity of our protein. Now we need to isolate our protein from the E. coli. The cells can be broken open, or lysed, through chemical or physical approaches. Then we can use centrifugation to make the E. coli cell parts form a pellet while our protein is in the supernatant. However, all the proteins from the E. coli cytoplasm will be in that supernatant as well. We can separate our protein from the others using chromatography. In chromatography, we pass a liquid solution through a stationary material. The stationary phase will interact differently with the various components of the solution, causing them to move at different speeds. Take this column. It is filled with tiny beads, and these beads can have a variety of different properties. Ion exchange chromatography involves beads with positive or negative charge. If the beads have a positive charge, proteins that also have a net positive charge will repel and move quickly through the column. But negatively charged proteins will be attracted to the beads and move much slower, allowing us to separate the proteins in the mixture by charge. In size exclusion chromatography, the beads have tiny pores in them. Smaller proteins get temporarily stuck in the pores and are slowed down, while larger proteins quickly flow around the beads. This allows us to separate the mixture by size. Thanks to the XPC ProParam tool, we know the size and charge of our protein and can predict how it will behave in these columns. But we're actually going to use another method called affinity chromatography. In this approach, the beads will very specifically interact with the one protein we care about. We've actually modified our protein to make this possible. Recall that between the start codon and the coding sequence of our gene is a series of histidines, a his tag. Our protein of interest is fused to this sequence, and a series of consecutive histidines has a strong affinity for nickel ions. So if we attach nickel ions to these beads, our his-tagged protein will strongly bind to them. No naturally occurring E. coli protein has a bunch of histidine amino acids in a row, so they'll pass right through the column. After that's happened, we can add the chemical imidazole to disrupt the interaction between the protein and the nickel, eluting our protein, purified from all the other E. coli junk. One potential drawback of the his tag is that it could interfere with the protein's normal function. But there is also this factor XA site between the his tag and the coding sequence. Factor XA is a protease that cleaves proteins at this amino acid sequence. So if we mix it with our sample, it will separate the his tag from the main protein. Now we have our protein of interest and can use it to perform any sort of biochemical assay we want. If your protein is an enzyme, you can perform enzymatic assays similar to what we've done earlier. If you're curious about what your protein interacts with, there are binding assays. You can learn about its structure with protein crystallography. And making purified protein is only part of the power of cloning. Our plasmid is specifically designed for E. coli, but we could modify some of the features so that the plasmid propagates in mammalian cells, and a mammalian RNA polymerase will transcribe the inserted gene. Now the gene can be expressed in cell lines that match the original host. And instead of the his tag, you could fuse your protein to any other amino acid sequence you want. For example, there are proteins such as GFP that are fluorescent. If you fuse your protein of interest to a fluorescent protein, you can use fluorescence microscopy to find out where your protein is localized in the cell. Cloning allows for so many possibilities. In lab next week, your group will research a technique that will help you address a question about your protein of interest. And in the last week of lab, you'll present your proposed experiment to the class. That does it for Bio43 pre-lab videos. Bye!